Hello everyone, Happy New Year. Today I have the pleasure to be with Nahuel Policelli. Hi Nahuel.
Hi Camille, how are you?
Very good, thank you. Nahuel is a postdoc at Boston University, and today he's going to tell us about his recent paper, native and non-native trees can find compatible mycorrhizal partners in each other's dominated area that you just published in soil. Okay. Great. So, Nahuel, can you tell us a little bit about the system of plant invasion and the role of ectomycorrhizal fungi?
Sure, yeah. Well, the thing is that most plant species need mycorrhizal fungi to survive. So some of them, mostly the tree species, associate with a certain group of fungi that are called ectomycorrhizal fungi. And most of them are obligate symbionts with them. So the thing is that they need them to thrive and survive. And they need them to get nutrients and water from the soil in exchange of plant carbon. So they need them in their native range, but they also need them when they are moved to new areas, to new ranges. So you know that several tree species are moved from one place to another for different reasons, maybe for forestry or for production, and some of them become invasive. But one good example of that are pine trees. As you know, pine trees are highly invasive in all the southern hemisphere, and they are non-native in the southern hemisphere, and they have been widely introduced in the southern hemisphere. And some species of pine trees have invaded the southern hemisphere. But they need these ectomycorrhizal fungi also to thrive and survive in the new range. So if they don't find them, they cannot invade. So for example, for pine trees, the idea is that most of the ectomycorrhizal fungi have been co-introduced with pine trees, which means that they have been brought together at the same time or at different times, and from the same place or from different places, with the pine trees.
I think that in Chile, they first start to do pine plantation based on seeds, right? And it didn't work. Exactly. And it's only when they introduced seedlings, seedlings with the mycorrhizal fungi in the plantation, that they could start to have plantations.
That happened in Chile, and that happened also in the Argentinian part of Patagonia, in which in the 1940s, they introduced pine trees, as you said, only with the seeds, and they have to wait for the soil to be introduced so that the plantation can work. And at that moment, they didn't know exactly why that was happening, but then we realized that it was because of the absence of ectomycorrhizal fungi in during the first introductions. So once the co-invasive fungal symbiont is present, pine trees and other plant species can then invade. There are other examples in which the plant tree can use not co-invasive symbionts, but symbionts that are already present in the native range. Or they can even change the community in a way that their invasion is favored. But all these below ground aspects of invasions are generally overlooked compared to the above ground aspects. So that's why we were really interested in what was going on with plant invasions in a below ground aspect.
Okay, and so in your study, you studied pine invasion in a native, was it Nothofagus and Araucaria system, right?
Yeah, these Chilean researchers that we shared the paper with, they have an amazing group of people, and they also have these amazing places in which you can actually see the pine invasion in a kind of a perfect gradient. So you can see the plantation and then the mature invasion and the invasion front over sometimes grasslands, but most of the times over Nothofagus stands. You know that the native trees from that area are mostly Nothofagus, and those are the only ectomycorrhizal trees that we have there in Patagonia. So the interesting part of that invasion from the below ground aspect is that you get non-native pine trees invading over a Nothofagus forest, but both hosts are ectomycorrhizal. So Nothofagus associated with native ectomycorrhizal fungi, and pine trees associated with the non-native co-introduced ectomycorrhizal fungi. So the main question was what's going on below ground.
And so can you tell us a little bit about this experiment that you made with this system?
Sure. So we took soil cores and roots from the dense invaded areas, not the plantation, but the mature invasions in which you can see like very dense pine stands with adult trees growing. And we also took soil cores from the non-invaded stands, where you get Nothofagus as a dominant tree species. So we took these soil cores from those two different locations, and we actually planted seeds, sterile seeds from pine trees, Pinus contorta and Nothofagus antarctica in all the possible combinations. And we also used sterile controls, like sterile soil, as a control to compare the growth and the percent colonization of the roots.
And so, surprisingly, somehow, you found that both pine and Nothofagus seedlings grow equally well on the two type soils. Exactly. So what was your explanation for this? Were you expecting to find these results?
No. We were expecting actually to find pine trees growing less in the Nothofagus soil stems and the same with Nothofagus. But we found that, as you said before, we found like equally growth for both hosts in both soil types. And not only growth in terms of biomass, but also we found equal percent colonization of the roots. So when we took the roots out of the soil, we noticed that for pine trees and for Nothofagus trees, both were highly colonized, but we didn't know the identity of the species that were colonizing, because it can be like all native, a mixture of native and non-native ectomycorrhizal fungi, or other situations. So we wanted to address that, and we wanted to identify the species of ectomycorrhizal fungi colonizing the root tips. So that's why we perform molecular analysis and sequencing to know which species were there. And one other thing that we didn't expect is that we found pine-specific ectomycorrhizal fungi in the native stands. So we found particularly pseudoid fungi, which are known to be heavily invasive by themselves because they disperse independently from the pine, and they can reach very long distances from the initial introduction point. So we found those highly invasive fungi in areas without their hosts. So this famous paper from Collier and Vidar-Tondo from 2009 called Waiting for Fungi. Well, here we had like waiting for hosts. So we had this fungi waiting for their host to arrive so they can co-invade the Nothofagus stands. And we also found Nothofagus doing well in the pine stands and actually associating with their own native symbionts that were in the pine invaded stands also. So those were two unexpected but very interesting results we got.
So do you think that this invasion of exotic ectomycorrhizal fungi in a native stand, do you think so this is a distance and time related thing?
Yeah, actually there is certainly a cumulative time effect. In which like the older you get, the most spores you get also, even in like distant places. And the thing is that for Patagonia, at least, that is the system that I know best, is that you get like really like a lot of places with pine plantations. So we might have places that are considered non-invaded by pines for sure, like Nothofagus forest even, that are actually kind of preconditioned for the invasion by the presence of these highly invasive fungi. And they are not only dispersed by wind, they are highly invasive because they can also be dispersed by mammals, for example. And mammals can travel even 12 kilometers a day. So of course, if you get like really, really far to isolated places without mammals, without humans, and this is a spoiler alert, we are working on a paper in which we actually found these fungi in really isolated native stands in the lake, in the middle of the lakes without pines. We found some invasive ectomycorrhizal right there. here.
Oh,
And
wow.
we didn't get non-native mammals. So the thing is that they kind of, this invasive fungi, and we have to start considering fungi by themselves invasive, are conditioning native forests. So yeah, there is an effect of time and of distance, but we have to be aware of this situation.
Of this, yeah. But at the same time, somehow it seems that even in the pine plantation, the native ectomycorrhizal fungi, probably for example in the spore bank, they are able to maintain. So that would be kind of a good news if you want to do restoration ecology and restore these native forests that have been perturbated by plantation, because these fungi, they're still there.
Yeah, I agree. Right? I totally agree, because and probably in those sites, in when you get where you get like really dense pine invasions, you can see like really old Nothofagus host growing in the middle of the invasion. So very isolated, but still there. So those hosts may be really important as a source of also ectomycorrhizal fungi mycelia that might be colonizing, and also spores that might be colonizing other hosts nearby. So when you, a big problem when you, for example, do clear cuts of pine trees is that spores from this ectomycorrhizal, invasive ectomycorrhizal fungi actually can stay in the soil for years, and it's really hard to eradicate them. I mean, the invasive ectomycorrhizal fungi. So the presence of these native ones might be important for when considering, like, for example, soil translocation or the source of inocula for new trees establishing in those areas, native trees.
So you recently published another really nice paper about this question of how to include mycorrhizal fungi in restoration. So what would you suggest to try to include these aspects?
Well, actually, yeah, that paper that it's published in Frontiers in Forest and Global Change, it had this applied perspective that I really liked, in which they asked us to write about this restoration purposes, in which it's actually underrepresented in restoration. I mean, recognizing that temperate and boreal forests are really, I mean, all of the trees are ectomycorrhizal. When you go to the literature and try to see if they have been considered in restoration purposes, you get kind of disappointed. There are some examples from Spain and from other places where they have been considered, but they're really underrepresented. So for Nothofagus forest, they for sure need to be considered, I mean, native ectomycorrhizal fungi. And they need to be considered when planting trees from nurseries, for example, because if you don't get inoculation of Nothofagus from nurseries, you will not get it then on the field. And also, if you get in nurseries, that doesn't mean that you will get successful inoculation in the field, because the communities might change. So we don't know much about it, but we for sure need to consider that. And also for pine trees, the interesting part is that for sites in which pine trees are native, for example, here in the US, these highly invasive ectomycorrhizal fungi that are highly invasive in the southern hemisphere should be considered for restoring pine stands here in their native place, because they are really well at colonizing trees, and they are really resistant to disturbance, to soil disturbance, even to fire, some of them. So for native pine forest, the information of this pine invasive stands in terms of their ectomycorrhizal fungi should be also considered for restoration purposes, I think.
Yeah, that's a good point. I didn't think about it, because there's exotic species in the native range. They're not necessarily the dominant species in this pine range. Do you know? Ah, no. Okay.
I mean, the thing is that, for example, these invasive sulet fungi that are heavily invasive, and they actually can colonize 100% of the root in the invasive place, actually represent less than 5% of the root colonization in the native place. So they are not that well represented in their native place. But they are good at, for example, early disturbed areas in which you get a fire, and then that's when you get like the most of these invasive fungi proliferating and colonizing new roots. So that's the point in which you should focus your restoration efforts on.
So you mostly studied the dominant ECM fungi that were associated with the seedlings. So I was wondering if you do an analysis of the general ectomycorrhizal community that you find in the pines and in the nothofagus, do you think you would still find this separation, or do you would find more ECM fungi that actually are able to associate with both hosts?
Yeah, I would love to do that. Like looking for this not that common EMF species, because of course with this paper in plant and soil we cannot rule out the possibility of like co-sharing, like sharing ectomycorrhizal fungi symbionts. There are also other papers that even your papers that report, for example, Amanita muscaria in nothofagus roots, and we don't know the exact function of that. What's going on with functioning of that system, but for sure for that like less common species we might get more similarities in the community. And in the case of pine trees and nothofagus, they're really far away phylogenetically, so it might be more important for more related plant species in which they can actually use the ectomycorrhizal fungi of the native host to invade. So they can actually get there and use the native community of ectomycorrhizal fungi and actually invade this new place. And there are some examples, even in Patagonia, not in the forest, but in the steppe, for example, of those cases. So I think that would be a really interesting question to address for those like close related species too, for these rare species.
Yeah. Great, thank you very much. That's really interesting. So do you want to tell us a little bit what you're doing now that you are in the Northern Hemisphere, and what are your plans for the future?
Sure, yeah. So I'm working here as in the second year of my postdoc at Boston University with Jenny Bandnagar and her group too. And actually, I'm still working with these highly invasive ectomycorrhizal fungi and their interaction with pine trees, but I'm addressing new questions in terms of their relationship, their interactions with the rest of the community, the soil community, so the saprotrophs that live there, and how these highly invasive ectomycorrhizal fungi with the plant, with the pine tree, interact with saprotrophs, and how that have impact in the soil biogeochemistry and, for example, in the CO2 release from soil, depending on the carbon, the carbon that is in the soil and the carbon that is in the plant. So we are evaluating different carbon and nitrogen conditions in the soil and in the plant, and see how that interactions between ectomycorrhizal fungi and other fungi, free-living fungi, can affect the biogeochemistry of the soil. So I'm interested also in comparing Northern Hemisphere situation with invasions in the South Hemisphere. It's hard to get seeds and seedlings and everything, like transport them, of course, but we are making our big efforts to get like this common garden experiments with that type of questions too. And for the future, yeah, there's these new ecosystems and these new invasion scenarios, like for example in the steppe, not only with pine trees but other ectomycorrhizal hosts. Would be amazing and interesting for the future. So
awesome. Well, thank you very much, Nahuel, for your time. And we will be back soon with more interview for the South American Mycorrhizal Research Network. Bye.
Thank you. Bye-bye.